Our session was hi-fi emulations. This is as opposed to lo-fi emulations, and aiming at whole brain emulation with connectomics. So what did this actually mean? First, we tried to define that a little bit, which is like making. It's kind of runs a spectrum, like making real digital people, aka uploading, run them really fast, upgradable, or you could have like subhuman, like hi-fi M, which is still useful, sort of like parts of a person, maybe specific parts of brains. But the main focus is connectomics, sort of the structure of how neurons are connected, and then some kind of structure to function translator. Should be like here, and then okay, something like that. And then, yeah, computation and virtual environment, and then some fundraising strategies. Mostly we focused on these first two, connectomics and structure to function. Cool, we got some links already. So maybe I'll skip over the structure to function for now, and maybe just talk about the connectomics. Here we go. Cool, so the current state of the art is electron microscopy. This is time-consuming, especially putting it back together, but using this is how like the Flywire brain was put together, and the like cubic millimeter human brain and mouse brain. On the horizon or very near is expansion microscopy, and this is but there's no proven high throughput pipeline. Got like 5 minutes to go through. I won't read it out for you, but expansion microscopy is sort of like it's like in the wings. It's really close. There's like expectation that this is going to be the new like connectome mapping technique using optics rather than electrons, and you expand the tissue so that you can the wavelength of light can still resolve features. Weird new approaches are something like well, actually, multi-beam is already a thing. But adding barcodes to expansion so that you can trace neurons across like cut lines and never be confused about where neurons go. Yeah, weird like expansion X-ray microscopy, microtomography. Yeah, and and then other weird ones too, like ensome. Like no one's ever used before, just an idea, and like using semiconductor like polishing kind of things, and then other ways of like labeling things for electron microscopy using shapes, shapes of proteins and things.
Yeah, genetically encoded proteins that form shapes that you can segment and say this is this shape, this is this shape, and so it's fun.
Yeah, and then. Technical bottlenecks to success. Well, I mean, you know, the brain is just like a vast amount of information. It's not also clear what parts to label. So as you're getting a structural connectome, what things can you label? You know, channels and neuromodulator receptors and a bunch of other things. And also you can label everything in one cell or in bulk, and then try to like while you're simulating it, then remove those things to see if you can still like predict the function, and then try to like shrink the number of things you actually have to label. Some ideas, and then let's see. Do you want to say anything about tracing?
And with regards to the trade-offs, I definitely would recommend the preprint that I put out recently with Randall, because that definitely covers a lot of the trade-offs in a fairly thorough way, especially with regards to speed and resolution.
Yeah, that's that. Okay, other last quick things to mention. Yeah, so okay, so how much is this going to cost, at least for the connectome? We've got the like very optimistic estimate from me, and the sort of more realistic estimate from Andrew. So somewhere between like 2 to 5 years to get a mouse, and 5 to 15 years to get a human, like single neuron kind of connectome. Now, can you actually run that? Yeah, TBD. I was pricing something like 30 million to 200 million to get a mouse, and 50 to 400 million to get to make it functional. And then, yeah, I'm not sure if we did that for human, but I'm estimating somewhere like billion or 2 or 3 or 4 to get to human. What's a billion here too?
Yeah.
Whatever.
I mean, I think a lot of that cost comes from the post imaging as well, because some of the newer imaging techniques, like what E11 is doing and the X-ray microtomography stuff I've proposed, could get the cost down for the imaging part, but the reconstruction and segmentation and computational processing and storage even might be more costly at this point.
Yeah. Okay, and then last thing to kind of close on is one, choosing a specific microscopy technique. I think the top two are probably the real ones to go with, like EM and expansion microscopy. But like, why hi-fi rather than lo-fi, which is next? I mean, because it's way harder and way more expensive. And so mainly the reason is like safer. It's like much more similar to like what we are. And but it's a spectrum. And so maybe lo-fi could be made safe enough. But the hi-fi path also would rely on a bunch of the lo-fi recording as well to kind of make the single neuron kind of models and dynamics like more realistic and not like go off the rails when you start going to larger length scales. That's it.
All right, thank you, everyone.
Well, stay right here. We have questions.
All right.
Yes, thanks. Thanks so much. Just a very quick question about error robustability. Sorry. How wrong can you be for these models to not work? Like if you have 0.1 error in something, like your proofreading and all these things, do you have a sense of that? Do you guys have a way to quantify how much off can this method be so that it gives you the main things you're looking for at the end of the day? Because it sounds like there's obviously, like in all methods, there's some places where that we just can't eliminate errors.
Yeah. I'm gonna point at like Phil way in the back. How far off can we be? Like,
Shout it out, Phil.
what was the question again? So this is like how many like errors can we have in the tracing? Let's say. Yeah.
Yeah, I don't know if we have a great answer for that. I mean, I guess tracing is improving pretty dramatically with like so when I started this, I did manual tracing, which was a huge pain, and then things got better and better and better. And I mean, I don't have a good answer, but like I'm hopeful that it shouldn't be a major problem so long as the data quality is good enough.
Part of the barcoding is actually that it's like self-audibly like error correcting.
Oh, okay. You made your point.
That's it.
Get to you in a moment. To have that type of tracing super accuracy, because it'd be less easy to almost like stochastically correct it by using sort of slight algorithms that would almost like generatively fix the errors. But anyway, that's just sort of speculation. So Aurelia.
So I also don't know the answer, but I would look to a clinical experience of pre-Alzheimer's dementia to get some type of baseline. So you can there's videos on YouTube where you'll see individuals that, they'll say, well, I don't know, something maybe seems a little bit wrong here, so I thought I'd get it checked out. And that corresponds to like losing 10% of your brain mass. So that you can lose a lot of this stuff and you'll like start to kind of notice it, sort of.
It's fair enough, but the rest of the brain is working properly.
It doesn't mean that, the specific kinds of errors caused by tracing glitches are exactly comparable to that, but this is not like the most fragile type of thing ever.
And I think the errors caused, like I was sort of alluding to a moment ago, the errors caused by tracing, like merges and splits, could be corrected almost in a sort of estimate kind of way. So you would be able to train some kind of generative algorithm to sort of reproduce in those particular regions what it should probably look like. And I think that would probably be sufficient to resolve most of the minor errors that would be scattered throughout without disrupting the connectome too much in the big picture, since again the brain, as Aurelia mentioned, the brain is pretty plastic to a lot of changes, and so it would probably adapt, especially once you started the functional system.
Well, one more, and then
Might not be for this talk, but just in general, I'm somewhat curious, like what there might have been to be learned from, like, for instance, like the human brain projects, like successes and failures, and also like in terms of plans, like the ongoing work at the Allen Brain Institute, like in what ways could that be leveraged and for these efforts in terms of, like, maybe it's overkill, like Chris definitely he wants every neuron and every type, but yeah.
I definitely think that the Human Brain Project sort of has two big lessons. One is don't promise everyone that we're going to have a simulation of the human brain in 10 years. Be a lot more tentative about it, and don't make any like specific promises. Say, maybe we'll have the mouse brain by this range or something, but we don't know for sure. Don't just make some big flashy headline that everyone picks up in the press. Because then when it doesn't happen, people will be disappointed, and then you'll get a bunch of backlash. The other big lesson, I think, is we do actually need the connectome, at least to some degree, at least significantly more than what they did, which was sort of creating artificial, almost like applying that generative algorithm kind of thing that I mentioned, but just using it for the entire connectome and just creating something that looks like a connectome, but isn't really a connectome. And so they did that, and it didn't really work out very well. They got sort of vague statistical insights, but nothing more. And so I think that we do need the connectome, and that's the other big lesson from the Human Brain Project.
all right. Thank you, guys. Thanks a lot.